Good afternoon, everyone. Thank you, Bruce, for the introduction, and thank you, everyone, for having us here. I'll actually be talking about, instead of new food development as is written, is actually more about new laboratory development that is going on in KALRO. And this is a project that is in collaboration with all the people you see there, VIDO, University of Saskatchewan, and funded by IDRC. So it's running concurrently with what you heard presented earlier this morning by Andy Porter, the CBPP conjugate vaccine. But this aspect of it is actually to improve our laboratory facilities in Muguga in Kenya. So just a little bit of history. Muguga was founded in the late 40s, and different aspects of the institute that focus mainly on livestock diseases were developed in Kabete and in Muguga, where we are based, which is approximately 12 to 17 kilometers out of Nairobi town. This allows it to be a facility where we can do particular diseases that need quarantine facilities, basically, and also to have a lot of space because we handle live animals. So Muguga has been in the research field for livestock diseases that are very relevant to African countries such as rinderpest from the early 60s, and they've continued over the years to contribute to other diseases such as ECF and CBPP and CCPP, and even the current research that we see today that was also discussed earlier, which is on CBPP. Our main focus is not only on bacterial diseases, but we also do some virology and some protozoology, hence the crossover of diseases. So this is the facility, how it looks like. When you come in, this is what will greet you at the gate with our logo. This is our main laboratory facility that was the initial facility, and this is where we used to be based, the bacteriology laboratory. With time, we've outgrown this and expanded. And so some of the discussions that you've heard is also how we can upscale and improve production, whether it's of recombinant proteins, whether it's in terms of high throughput systems to do more samples, especially when it comes to clinical trials. So that's one of the things that brought up the discussion. So a few more pictures. We have qualified animal veterinary staff that handle animals, and we do handle live animals, as I mentioned. We do this internally also and sometimes outside, so we have space to do research and clinical trials inside and outside. And as I've just mentioned, as we know, diagnostics is very vital in monitoring of diseases, and this helps in improving affordability of treatment, especially in our setting in Africa, where from a laboratory facility to the field is usually quite a distance. So the discussion did come up on how we could improve accuracy, speed, and affordability of disease detection. And we thought of two systems that would allow us to do this. This is the Evolis and the Bioplex. So before, we had all this land. Muguga is based on 999 hectares. As I mentioned, this is to allow quarantine, this is to allow enough facilities for animals to move around and graze. And so we had land, and we already had the expertise in terms of we were still doing manual diagnostic methods such as CFT for CBP. And you can imagine the cost of that, especially with the current method where you use antigens that the batch is often varied. This is also time consuming from not only running the test, but also obtaining samples. And with the help of collaborations with the University of Saskatchewan and VIDO, and also IDRC, we were able to establish this new facility that you see here. This is a laboratory now, and this is mainly for bacteriology and serology. It's mainly dedicated just to diagnostics and vaccine discovery. This is the Bioplex system, which we were very interested in obtaining, and we did get, and also the Evolis system. After we had these machines installed, we ensured we got, of course, ground support to, in case of any access for materials or even just warranty in terms of parts and product. And we also got our staff in to get fully trained. We had Maureen here, Eric, and two other staff actually even come to VIDO and get trained up on this. And we continue to make sure our staff on the ground are upgraded and trained. So this platform that we installed was not only to allow us to improve, reliability, the cost of running diagnostic tests, but also to help us expand into the number of tests that we can actually detect from field samples. Now, just a little bit about the bioplex, sorry. The bioplex is actually a multiplex assay. This allows it to run 50 to 100 analytes in one well. The wells are actually 96 well. It's a 96 well ELISA format. So it can allow you to run up to 80 samples, depending on how many controls you want to have. So you can imagine that's 80 samples by 50 diseases, yeah? And you can detect in one sample 50 to 100 diseases for a serosample. You can imagine the power of that. And this is in as little a volume as 12 microliters. You can imagine if you had 80 samples that you wanted to detect, and perhaps you wanted to detect up to 2,000. This, if you think about it and you do this in a laboratory with manual ELISA assays, this could take you up to 60 hours if you're doing it continuously, slaving away in your doctoral. But if you do this with this new bioplex system that we've gotten, you will get results within 3 hours. These 3 hours can be reproduced within the same day, the next day, to ensure there's no variability, with minimal samples. So the power of that for us was that now not only can we detect CBPP in one serosample from a cattle in as little volume as 12 microliters, but we could also detect 50 to another 100 diseases, depending on if we had the analytes. That is the most crucial part of it. But you can imagine the cost that has brought down in terms of reagent volumes, in terms of time, and of course in terms of having to reproduce or repeat an assay. The other system we got in was the Evolis that I'd shown you earlier. This is basically an enzyme immunoassay, so it's also on an ELISA plate format and uses minimal volumes. It's a semi-automated system, so the beauty about that is it allows us to load samples directly after bleeding from vacutainers. So for some diseases that perhaps you want to minimize any contamination between animals or even to laboratory staff, this is very possible because you'll simply load your vacutainer after your serum is settled and put it in. And you'll run the same ELISA plate again for 3 different assays, detecting as up to 10 different antigens within 4 hours. So the reproducibility and also the handling of infectious samples from infectious diseases, was catered for with this other system. So currently, where are we? We've moved from actually not having that system in place. We previously did have, as I've mentioned, the other systems that we use, especially for CBPP in terms of CFTC, ELISA, but these are outdated methods. They also cost too much. So now we've moved from that system to having this system that we've currently got in place, and we are optimizing not only for CBPP, but also all the other up to 20 neglected African diseases that you've already had mentioned here, the pox viruses and others. We're also currently working now on developing protocols for those other diseases. We also want to implement good laboratory practices, and we're in collaboration with FAO to set the standards. They've been on site. They've checked with us with what we need to scale up on, improve on and implement. And we're also working towards joining a laboratory management integrated system in collaboration with FAO and an accredited laboratory in Italy who would install the system. This would allow us to become a reference laboratory in Africa for disease surveillance, but also for monitoring the effectiveness of vaccines and any other re-emerging diseases. With this, I have to say that all that machinery was possible through the funding of IDRC, and also the selection and discussions on what would be crucial long-term in terms of detection for diseases and also vaccine development was also possible because we're in collaboration with Saskatchewan University and VIDO. So I'd like to thank our collaborators for that. And also I'd like to thank the cattle, which of course are involved in allowing us to be able to do all these assays and, come up with good assays for diagnostics and vaccine research. Thank you.
Okay, thank you very much, Dr. Nimmo. Nicely presented. You presented your future plan is to have accreditation of the laboratory. It's very important. That's good also. I mean, I didn't see in your presentation, especially the biosafety, because when you are thinking about accreditation 17025, biosafety and also ethical things, because you thank the animals, but what is the ethical issues? Because you cannot do for cattle and bovine and other animals as simple as it is. There should be ethical regulation, how you are really tackling the ethical issues, especially for East Africa, because we want to learn from you. The last one is, I mean, chemicals and reagents, especially for PCR and other high-level instruments are very costly. I mean, how your plan to make sustainable, especially to produce basic chemicals and reagents made in Kenya. Thank you very much.
Thank you for that. Those are all relevant questions. So I'll start with ethical approval. We are a facility that has been running clinical trials for the last so many years, before I was born, as I've mentioned. So we do have a committee in place. There is an ethical committee in place, and this involves not only us as an institute, but it also has sitting on it government regulatory boards, and we have to conform to it. So any clinical trial we're about to run, we have to get approval and a clinical number allowing us to go ahead. With that comes the disposal of animals, like you're saying, animal welfare and handling, and general laboratory practices also. And we already have that in place currently. So that is all there. It's that in this situation is that we wanted to improve the diagnostic platform. As we go ahead and we make it sustainable, we will hope to improve the other facilities in terms of biohazard handling and what else we can handle. Yeah. But currently, we do have them in place. They're not as, you'd say, as new, but they cater for all pathogens. And finally, sorry, your last question was on reagents and PCR. So currently, yes, molecular research is quite expensive, that we agree. These are serological platforms, so that affords us that affordability, like you're saying. And some of them are semi-automated, so you can still make a few things. But we're trying to move away from that. So for the serological platform, there is some expenditure. And hopefully, as we continue to do disease surveillance for other people in Kenya, in Africa, and be a reference lab, we'll be able to sustain that cost and implement PCR into it, or the molecular costs that come with it of detection, yeah?
Yeah, thank you. My question is simple. You know, in Nairobi, we have ILRI, which has a very good facility. Peka, you must know. So I'm wondering whether they know about your work and you know about them, whether there's any really collaboration between the two institutions.
Yes. Thank you. So it's very interesting you should mention that. I wasn't going to mention it, but currently this platform in Africa does not exist. We are the only facility in Africa who can confidently say we have these two machines capable of actually delivering at this level, including the expertise trained to this level in terms of our whole team. According to the company who even supplied it to us, Biorad, we're the only two from north, up in Eritrea, all the way down to Mozambique, who have this platform. So Becca do not have this platform, Becca at ILRI, and we're in very good talks with them because they have a very good genomics platform. And so we don't need to replicate that. And as you've heard, the idea of Science Now is to collaborate so that we keep the cost down and the quality up. So we're in discussions with Becca. They do know we have it. And one of the things I didn't mention is long-term, we want to actually incorporate Becca, that we do the serological side of it in terms of vaccines and diagnostics, and they can do the genomic side of it. So we're in good discussions, and we are happy to continue discussing with anybody else who would be keen on this.
Thanks. You've got this new serological platform you're describing, and you said the CFT. You know, everyone knows the disadvantages of the CFT, but I quite like the CFT. I think it's a good test. I mean, you presumably produce a lot of data comparing the two tests, have you? Or is this in routine operation now?
This is what we're starting up now.
Right.
Yeah,
okay.
so recently we did have a CFT screen, and our protocols are up and running. So now we're screening again against serology, and we want to do it a couple of times. And as you know, it's CFT, you always have to have somebody who's very experienced in it. So we want to get a second person so that we get a second opinion. Yeah.
So how does it look now? I mean, have you got some preliminary data? Because this is, quite revolutionary, I think.
Not yet, but actually you're the first person who comes to mind when it comes to discussing those results. So we will get in touch for sure.